Hi everyone, my name is Luka and I'm one of the tutors from the Writing Center. In this video, I will show you how to write a lab report. The first part of every lab report is the exploration section. In the exploration section, first you're going to write your research question. An example research question is written here. Which color of light, blue, red, green, yellow, orange, or natural sunlight yields the fastest growth in common sunflower? And this is the genus and species name. This research question is written as a question. It gives insight and details really matter here. So make sure you address what your variables are and also specify as much as you can about what you're testing. Next part is the background information. In the background information, you should show your depth of knowledge. You should cite your sources. In biology, specifically in science, you write for your citations. You write the author's last name and year. You should have 5 or more credible references, and the background information should be 3/4 of a page. Writing more than that you might run out of space. So for example, for this specific research question, I might research more into what sunflowers are, maybe which wavelength of light do sunflowers prefer, and what kind of wavelengths of light are available in those different colors of lights. So make sure you tie in your research and tie in your variables into your background information. Then you should write your hypothesis. It's a good way to start your hypothesis by, it is expected that. You have several sentences to write what you want, and you must state the outcome of your experiment before you begin conducting the experiment. So, for example, I wrote, it is expected that the natural sunlight will allow faster growth of common sunflower. This may be due to the diversity of wavelengths available in natural sunlight. So as you can see, the first part is my hypothesis, and the second sentence is a sentence that should be justified by the background information, and this sentence gives more depth. Then material section. Material section is very important. It is and in the material section you should state what materials you use. So for example, if I were to use common sunflower seeds, I would say how many I used, the company, their genus and species. If you were to do a measurement tool, you could say that you're using 100 milliliters of graduated cylinder. And then if you're putting measurement tools under the material section, you need to add the uncertainty. So for the graduated cylinder, uncertainty is 0.5 plus or minus 0.5 milliliters. The materials that you use should state the uncertainty. However, if you're not sure, it is usually the half of the smallest quantity. So as you can see, those small tick marks on graduated cylinders, they'll represent 1. So you divide 1 by 2 and you get 0.5. Next section is the variables section, the variables table. And in this table, you need to state what your variables are. So for example, you have the independent variable, you have the control, dependent variable, and constants. This format is a great way to state your variables. In this column, you should identify the variables, and in this column, you should specifically state how you're going to control your variable. Next section is the methods or procedure section. This section is written best by step 1, step 2. However, you can put it in paragraph form. However, writing it in the step 1, step 2 form is more concise and it's more clear. So make sure you address safety. So obtaining safety goggles is a very important part of your experiment. Then make sure you specify the exact procedure that you're doing. How are you measuring what you're measuring? And then always include cleanup. So after the procedure comes the setup diagram. In the setup diagram, you only must illustrate how you're going to measure what you're measuring. So for example, as you can see, there's a ceramic pot, the flower, the plant is right here, and I'm going to use a ruler to measure the height, and I'm also indicating the light source. Make sure you do not take a picture of your materials, because that's not what setup diagram is. You must only take a picture of how you're going to measure what you're measuring. After that, you need to include the risk assessments. For the risk assessments, this is the area dedicated to the safety aspect of your experiment. So it is a good idea to include general safety, in which you may discuss if you're using any glassware, if you're using acid or other chemicals. And then you can also address the ethical issues, if there is any, in your experiment. After this comes your analysis section. In the analysis section, first, you must include your results. So for example, first I included the qualitative data table. In here, in this column, I labeled the IVs that I wrote observations for, and over here I wrote the observations. Next is your raw quantitative data table. In here, you will type in the raw data, the data that you actually measured. So this is a good format to put your data table in. However, it is all data tables are unique to the experiment, so yours may not look like this. However, you must include the trials. You must specify which variable the numbers correspond to, and you must always include the uncertainty. It is also a good idea to say table 1. This table illustrates, and then whatever you're putting in here will go here. Next is the process quantitative data table. This is where you may measure your averages, your standard deviations, and your t-tests. It is also a good idea to title this as table 2, so it's easier for you to discuss in the analysis section. In here, as you can see, I added the variable names and also specified them. And make sure you always add the units for the average. After the process data table comes the data processing accounting. In here, you must briefly explain how you process the data. You can mention Excel, so if you use Excel equations, but you must provide an example. If there's a certain formula that you used or something to show how you actually process the data. Then comes the graph. So for the graph, you must only graph process data. However, this is unique to each experiment. And also you must think about what type of graph you will need to use. Will it be a line graph? Is there like a correlation, like a linear correlation between your variables? Will you use a bar graph, a scar plot, a pie, which adds up to 100%, a histogram, or there's a lot of graphs that you may use. An example graph is down here. Your graph must always include the title up top. You must label your axes. The x-axis is your independent variable, and your y-axis is your dependent variable. In here, I used a bar graph, and as you can see, I also included error bars. Error bars are very important in your graph. You must always have error bars because there will be some error in your data. The error bars, most likely, you'll use your standard deviation for your error bars. Then comes the written analysis. In here, you must write one paragraph per variable. So you're going to have 7 variables total. You're going to have 6 paragraphs total, 5 independent variables and 1 control. In each paragraph, you will compare your IV to your control. Good things to do in this section is to discuss qualitative data, explain the numbers, both address raw and processed numbers. Talk about the trends on the graphs and what do they show. Evaluate the error bars and how they may impact your data. And you should also state the t-test values, but do not draw conclusions in the written analysis. Do not refute or accept hypotheses yet. The next section of a lab report is the evaluation section. This section includes both the conclusion and the evaluation. Your conclusion should be approximately 1 page. You should restate your hypothesis and restate your t-tests. And now you may state whether or not the hypothesis was accepted or rejected. And you may be able to draw this conclusion by just looking at the t-tests and their p-value. If p-value is greater than 0.05, you must refute your hypothesis. That shows that your IV did not lead to DV. You should also support your conclusion by qualitative data and your numerical data. It's a good idea to also connect to research from your background information section, and that's a way to justify your conclusion. Now, in the evaluation section, in here you will evaluate the strengths and weaknesses of your experiment. So first, evaluate the variables. How did you control the variables? Explicitly state them. For example, you can say all plants were equally exposed to their light. The next one can be biological uncertainties. What errors arose from your plant? For example, were there some that did not germinate? You should also state the instrumental uncertainties. For example, how did the uncertainty values impact your data? And did your original hypothesis, was it refuted because of the uncertainties? You should also address the strengths and weaknesses of your lab report, mostly focusing on areas of improvement, but also stating what you did good. Tie this into your procedure. Did you state everything you needed to state? Did you change anything during your experiment? And how would that have affected your results? So, for example, the strengths of a potential procedure for this experiment could have been some error was eliminated when all plants received equal amounts of their light. A weakness can be that the procedure may not have measured how to actually measure the height. So do you measure the natural height where you do not when you do not touch the plant and you just measure the height without bending the plant to measure it from like the tip to the bottom? And then you should also suggest some possible improvements. Your weaknesses should lead you to your proposal of the possible improvements. So, for example, the possible improvements of this experiment could have been to better specify how to measure the plant, and since the control was placed outside for natural sunlight, maybe think of some way to ensure no outside factor, for example, wind, wildlife, or rain, affects your results. This will conclude your lab report. I hope you enjoyed it, and I hope it was helpful.